Right, that's the empty tRNA. That's the deacylated one, right? Because it has already, because what happens is, these are the two tRNAs. This is in the P, this is in the A. So once they peptidyl transferase, this is an uncharged tRNA, but it's in the P site. This is a dipeptidyl RNA in the wrong side, which is the A side. So you need EFG, which translocates the ribosome. So now this is uncharged, leaves through the E site. This is dicharged, still in the P, and the A site will come in, another one comes in, and you repeat the process. Now there is one role that we didn't mention. If you have EF temperature unstable, you have EF temperature stable. What's the role of EFTS? To recycle EFTU. Because every time EFTU brings an amino acid, amino acyl tRNA, it gets essentially inactive. It needs to be recycled. That's the role of EFTS. So IF2 brings it in, EF2 brings the other one in, peptidyl transferase transfers them. It's the only RNA. AFG sponsors translocation. One final point here. True or false, translation occurs in the 5' to 3' direction. In other words, the messenger RNA is read starting at the 5' and continuing towards the 3'. Based on what I just said, absolutely. Remember, the beginning of the gene is the 5' end. The end of the gene is the 3'. So if a ribosome is to translate an mRNA, it has to start at the 5' end and has to go towards the 3'. And it's easy to remember that mRNA is made 5 to 3 and it's read by the ribosome 5 to 3. So it's made and read in the same. Open reading frames are pretty straightforward. Polysomes, same idea. You have many mRNA, one mRNA, many ribosomes. Coupling transcription and translation together because you don't have a nucleus. And then also, there are examples like it in the textbook. I might give you an mRNA and ask you to like I said, translate it. I'll give you the code. Or I could ask you simply how many amino acids are in that mRNA. So how do you do it? If you're given an mRNA and asked to determine the open reading frame, what do you look for? AUG. Clearly, you look for a start, which is AUG, and you keep counting 3 at a time until you hit to a stop. It might be the first one, it might be the second one, it might be the third open reading frame that's the longest. But you might have in the same mRNA different open reading frames. Don't worry about the protein modifications either. So, I'm sorry, no, worry about them. Only because I remember we talked about chaperones too. Right, so what are chaperones? Because we discussed them also in chapter 10. Chaperones, right, are protein complexes that help other proteins fold properly fold properly usually. Questions about... Okay, so 10 is a relatively short chapter. So I'll quickly go through the main concepts. And with molecular regulation, right off the bat, be able to differentiate between constitutive, inducible, repressible. Constitutive is always on. These are called housekeeping. So constitutive, these are your housekeeping, so they're pretty much always on. Inducible, repressible. Inducible, turned on, and repressible, turned off. Now, turned on by something, obviously, turned off by something. So in general, these would be things like you need all the time, right? So for example, polymerases, ribosomal RNA, the cell needs them to survive every minute, every second, every millisecond, whatever. These typically are catabolic proteins. These typically are anabolic. And the reason is fairly straightforward, right? If you are trying to break down lactose, but lactose is not around, why waste time and energy to make the enzymes that are not used? So it's only turned on when lactose is around. Repressible is the other way around. If suddenly you have an influx, or suddenly you have a huge amount of vitamin, huge amount of amino acid, why make more? You shut it down. If you know this basic idea, the rest actually would make much more sense. So let's start with the lac operon. We have the Z, the Y, the A gene. We have a promoter, we have an operator, right? And we have the LAC-I, which codes for the repressor. So please know the function, right? I does the repressor, encodes the repressor. P binds what? P binds the, P stands for what? Promoter, which binds the polymerase. O is the operator, which binds the repressor. Z makes beta-galactosidase, which breaks down lactose. Y makes permease, which brings lactose in. A makes the transacetylase, which to this day, we don't know what it does. In fact, it's not required. So to metabolize lactose, they only need Z and Y. How does the system work? And remember, think of the big picture. Lactose is not around. Let's start with scenario 1. Okay. Minus lactose. Lactose is not there. Does the system need to be on? No. How does the system shut itself? Well, that's what the repressor does. This I is always made. So think of it the way. This is the repressor. In fact, this is the repressor. Because I would need... This is the repressor. The repressor will bind to the operator, and therefore polymerase can't bind anymore. So there is no transcription, no proteins. Suddenly, lactose is around. So now you have plus lactose. What happens? Lactose gets in through a permease that's already there, although in minimum amounts. The lactose inside the cell gets converted to what? To allolactose, which is the inducer. Allolactose will bind, this is allolactose, binds to the repressor and falls off. Polymerase will bind, transcribe. Lactose will be metabolized dramatically, efficiently, robustly. That's the lac operon. Now you will get questions just like that, what is the role of this, what is the role of this? Or you might get an application question, if you will. So for example, if you're told that the promoter is mutated so that it no longer works, what would you expect the expression to be? High, low, all the time, high when lactose is around, low when it's not, or vice versa. If the promoter is not working, what do you think is going to happen? Sorry? It won't be transcribed no matter what. Why? Because if there is lactose, let's start without lactose. If there is no lactose, right, it's going to be a double whammy because the repressor is there and the promoter is bad. So you really can't transcribe. But what happens if there is lactose? Well, if there is lactose, lactose will bind to allolactose, to the repressor. Lactose, allolactose binds, it falls off. Now you expect the promoter to be accessible, but it's mutated. It's always off. Trip operon, pretty much the same idea. the trip operon is you still have a promoter, you still have an operator, but now you have 5 genes in reverse alphabetical order. E, D, C, B, A. And we have a repressor way back. This makes trip R, which is the repressor. Promoter binds the polymerase, operator binds the repressor. These make how many genes? 5. Not make, there are 5 genes. But the polycistronic message carries 5 polypeptides, but only how many enzymes? 3. So you make 5 polypeptides, 3 enzymes that ultimately take chorismic acid all the way to tryptophan. How does the system work? There is no tryptophan. Does the system need to be on? Absolutely. There's no tryptophan. This is geared to make tryptophan. How does it do that? This trip R is inactive on its own. It's inactive. It needs this to be active. This is inactive. It can't bind to the operator. If it can't bind to the operator, what can bind to the promoter? Polymerase. Makes the message, makes the enzymes, makes tryptophan. Tryptophan is suddenly shooting skyward. There's already gobs and gobs of tryptophan. How does the system shut off? Tryptophan is the co-repressor. It binds to this aporepressor, which is inactive. This is called aporepressor. Co-repressor, holorepressor. This is now suddenly active. It binds to the operator. The promoter is blocked. Transcription stops. Tryptophan level go down. Plus, it's going to be used in proteins as well. That's tryptophan.
So tryptophan binds to the repressor of the repressor gene?
Right, exactly. The repressor gene, TRPR protein, is inactive.
Mm-hmm.
It's only active when tryptophan is around. Well, when it binds to it, not just around. Questions about?
Trip-L is just in front of.
Trip-L is the leader, right? So, frankly, you don't have to know because it's much more complicated than this. It plays a role in attenuation. But really, there's a big overlap between all of these. So Trip-L essentially falls in these two because the operator, there's many operators, not just one. So the operator is actually several sequences. So it is a little bit more, in fact, much more complicated. We talked about also sigma H, which is the heat shock genes. Again, know the big idea. You don't have to know the exact details. We also talked about anti-sigma factors, right? And anti-sigma factors. And again, we're not making this up. There are sigma factors, there are anti-sigma factors, and there are anti-sigma factors. That's the smallest chapter, maybe 6 to 8 questions or so, but nothing more complicated than what I just described. What does LAC-Z do? What does TRPR do? What do anti-sigma factors do? True or false? A vitamin-producing operon is expected to be repressible. True, because it's biosynthetic, repressible. To break down the sugar arabinose, let's say, you expect the operon to be inducible. True. If arabinose is there, it breaks it down. Inducible. Okay, 9 is another big one, and there's 2 parts to 9. Remember, there's mutations and there is gene transfer. So let me walk you through, go through the discussion by flow charting our way through it. So let's start with mutations, right? The mutation part. We can divide it at the DNA level and at the protein level. At the DNA level, you only have 2 types, right? You have transversion, transversion. At the protein level, you have several types. You can have silent, you can have missense, you can have nonsense. And remember, these are point mutations, mind you, right? Here we're talking about point mutations, not frame shift. Point mutations. So very quickly, what is a transition? Purine to purine, pyrimidine to another. What is transversion? Think of it this way. Transversion is like reversion, rhymes with it. So you're moving them around. You're going from purine to pyrimidine and vice versa. Silent does not change the amino acid. Missense changes it. Nonsense changes it to a stop codon. Right? Missense can be neutral if you have an equivalent amino acid. Positive, positive, negative, negative, big, big. What property of the code gives you silent mutations? What genetic code property? The degeneracy, absolutely. If the fact that you can change several amino acids, several codons and not change the amino acid, that's silent mutation. Questions about this? That's 1 or 2 points right there. What is a frame shift? Add, delete. Changing the whole reading frame. True or false? A 3-base addition is likely to be less problematic than a 2-base addition. Well, you would expect it, because if you add 3, the reading frame does not shift. Essentially, it will catch up, because you're adding essentially 1 amino acid. So adding 1 amino acid technically might be less, but again, I'll phrase it in such a way that there would be 1 clear answer, let's put it this way. Questions about, well, you have to know the difference between forward and reverse mutations, right? Forward will give you a mutant, reverse will give you the wild type. Spontaneous induced, yes.
Can you, you had a quicker question, and I think I'm still kind of confused by it, because you said it had to do with the forward and reverse, and it had to do with the phenotype.
Right. Basically, when you do, let's say this is your mRNA, and it's going to give you a protein that will fold this way. That's a weird, let's make it fold easier to explain. And let's say the protein just folds this way. So if you have a forward mutation, that means you knocked off the DNA here, which corresponded to an mRNA mutant here, which corresponded to an amino acid here. So now the protein opens up and it doesn't really close. Does that make sense? Okay. So if you have a reverse mutant, essentially you could have a mutation right there, which now is right here. So think of it this way. This used to be a minus, this used to be a plus, because they have to bond. So the first mutant converted this to a plus, now it got repelled. So the second mutant, in essence, could convert this to a minus, and now they come back together. So what did you do? You reverted the phenotype, but not the genotype. In fact, the mutant, now you have a double mutant. So for it to revert the true genotype, what has to happen? This very same base has to be knocked back, changed back again.
So in the question, the reason, because I know I got that question wrong, because I said the phenotype would be the same, because it's a reverse, and it's back to basically the same thing. Or because it's like you said, the plus or minus, or...
Well, right, the question said not necessarily the genotype. That's how it was phrased. Right. Okay, so spontaneous errors in DNA polymerase, talked about a reactive oxygen species, or it could be mutagens. You will have 2 or 3 questions on mutagens, if not more. Let's quickly flowchart our way through them. Mutagens, they could be chemical, they could be physical, they could be biological, right? I think we started with physical. Chemical of 3 types. We talked about base analogs, base modifiers, and intercalating agents. You can look them up. Please know the examples. Base analogs, we talked about 5, bromo, uracil. Base modifiers, we talked about nitrous acid. And intercalating agents, I talked about ethidium bromide. All of these do what? What type of mutations do they cause? Point mutations. That's one or two questions right there. Physical, real quick. What are the two examples? Ionizing radiation and non-ionizing radiation. What does each do? Ionizing example are X-rays. These break the DNA. And non-ionizing is UV light, pyrimidine dimers. Biological are transposons. And what do the transposons do? They basically cause frame shift mutations. That's another one or 2 points. Did you have a question?
Yes, I thought that the intercalating caused frame shifts because they inserted the
Oh, I'm sorry. Yeah, my bad. Thank you. Right, these two. Thank you. These two cause point. This causes frame shift. Thank you. Because it inserts itself and the polymerase can't really tell. I guess I went over it too fast. Questions about these? Okay, so let's do the same thing with the second part. The second part is gene transfer. I mean, one question could be, what is the difference between horizontal and vertical gene transfer? Remember, horizontal is across to unrelated organisms. Vertical is from mother to daughter cells, from parent to offspring. Right? Makes sense. Vertical, when you draw a family tree, essentially you're talking about the vertical transmission. When you're going horizontal, you're talking about unrelated transfer. It's called horizontal gene transfer. In bacteria, there are 3 types. There is transformation, transduction, conjugation. Transformation, 2 types. And again, I think, to be honest, I mean, it still helps me remember things. And usually, the more you write things, the more you remember them, because you're using more senses. So this could be an efficient way to organize and conceptualize all the information that you've learned. Transformation of 2 types. You have natural, that's you, and artificial. Natural, we also talked about 2 types, right? With natural, we talked about a strep pneumoniae and Haemophilus influenzae, right? Haemophilus influenzae. So very briefly, you're not going to again be asked in more detail than I'm just going over. So what are the hallmarks of these 2? How can you differentiate them? Because that's what the questions will likely be. The strep pneumonia model is a gram-positive coccus. The gram-negative rod model, if you will, is this. So this is a gram-positive rod, coccus. This is a gram-negative rod. What are the big differences? Strep pneumonia binds DNA what? Indiscriminately. This one binds DNA specifically. This one brings in 2 strands, but breaks one. This one brings in both strands. It's in the notes. This one will recombine or break things depending on whether they're homologous or not. This one will recombine because it's bringing in its own DNA. Right? So non-specific, specific, brings 1 strand, brings both, may recombine or degrade, typically recombines. Artificial, we talked about 2 examples, heat shock and electroporation. You don't have to know the details, electroporation. You don't have to know the details, but do know that you're either going to heat them or shock them to open up to allow the DNA to come in. Transduction, viral genes. Through viruses, you don't have to know the details because we didn't discuss them. Conjugation, there's a bunch of information here, starting with plasmids, be able to highlight the main types. We have resistance plasmids, we have resistance, we have bacteriocinogenic, metabolic, virulence, and fertility. If we've given examples, please remember them, right? So for example, resistance, obviously antibiotic resistance, bacteriocinogenic, code for bacteriocins, which are proteins that are produced against closely related species. So proteins by bacteria against closely related. Metabolic, break things down. Virulence, cause disease. PXO1 of Bacillus anthracis, PXO2, the ant plasmids of E. coli. And then fertility is the one that we spend a good time on. And that's what I'm going to finish with. In fact, can you backtrack a second? You do have to know the characteristics of plasmids. So, characteristics. Let me just... Very quickly, they're not always circular, they tend to be. They're found in eukaryotes and prokaryotes, right? They are either relaxed or stringent. They can be episomes, meaning they can jump in and out of the chromosome, right? True or false, they are essential for survival. False, because they could be lost. How can you be essential if you lose it and you still, the cell is still alive? Right. Yes.
Could you say that they're essential for resistance, or do they?
No, you could argue that too. You could argue that they are essential for survival in certain situations.
No,
but
not survival, but just resistance, like the trait of being resistant.
No, because in many cases, drug resistance is on the chromosome. Right. So it's not absolutely on the plasmid. It tends to be on the plasmid, but it's not always.
If it is, could it just be that it's an HFR?
Right, exactly. That's what we're going to finish with. Right. So that's what allows them to be transferred, if they are on the HFR. Okay, with conjugation, guys, let me quickly walk you through. It should be freshest in the mind because that's what we covered yesterday. But with conjugation, you have to be able to know the difference between an F plus and an F minus, an HFR and an F prime. Right? So, let's start with F plus. F plus is a male because it has an unintegrated F plasmid. So if you are looking at an F plus, basically the cell would have the big chromosome and it will have a short, small plasmid. That's the F plus. The F minus will have the same, essentially, size. It will have the same chromosome, but it will not have the F plasmid. In fact, let me draw it big enough for us to transfer. This can mate with this. Very quickly, it forms a pilus, brings it together, cuts. In fact, remember, it's two strands, right? So it nicks it at ODT, transfers one strand here, the other one strand replicated rolling circle. So at the end of the run, you have 2F plus. But know what? What was not transferred? Chromosomal genes. There was no blue genes transferred because it can't. It's not in the chromosome. Right.
Did you say that in the last lecture that I don't know if it was last lecture, but it doesn't go actually to the pill line?
Right.
So it's just bringing it together and then that.
Well, right, because as soon as the pili shrinks, it's basically just a fusion of the two cell walls, basically. Let's go back to F minus. This F plus can turn into this. The chromosome jumps in. Now it's no longer HF plus, it's F plus, it's HFR. It can still mate with an F minus. it nicks, it transfers part of it. The problem is, think of it this way, for genes A, B, C, D, E, F, G, H, whatever, for the whole chromosome to transfer, it's too long a time. So typically, part of the genes on the plasmid get in, and maybe A, B, C. So you're going to get essentially only this. So let's call these A, B, C. Well, this is going to be F minus. It can't turn into an F plus. But what it does is that it picked up A, B, C genes. So at the end of the run, you transferred chromosomal genes, whether they're resistance or whatever, but you did not change the sex of the person. That's the second one. The third one is what happens if you have an HFR and instead of going back to F plus, the red came out and picked up A by mistake. So basically, you're going to get something that looks like this. The red did not come out on its own. It picked up gene A and left part of it behind. This is not F plus, because in F plus you'd be all red, all blue. It's not HFR because it's not one piece. We have to give it a new name, and the new name is F prime. This F prime can mate with the F minus. This is small enough to be transferred on its entirety. So basically, look what happens. This cell already had an A here. It's a merodiploid. So this one does both. It transfers, in fact, does everything. It transfers plasmids, it transfers chromosome, and it changes the sex. And creates a stable merodiploid. It does everything.
I have a question. For the HFR, I think, I guess, in balance up with the F minus, if they actually put all the chromosomes into the F minus, would that change the sex?
That would potentially change the sex, but it never, right, because again, it takes 100 minutes. That's a long time. But right, I mean, in theory it would.
You say it changes the sex, so what is it changes
To F prime.
Oh, okay.
Remember, F prime is a male too. Right? In fact, HFR is technically a male too. That's the gram-negative model. In gram-positives, we only talked about Enterococcus. Does not use pili, uses pheromones and aggregating binding substances. Nothing beyond that. And I think there was restriction modification. Was that the last one? Don't have the note. I think the restriction modification was the last one. Don't have the notes for chapter 9. I think restriction modification. Oh, microbiology, TI plasma. Right. There was two quick concepts that are two parts. One was transfer across plant cells, Agrobacterium tumefaciens. That's the one that has the TI plasmid and causes crown gall. That's the extent that you have to know. Agrobacterium causing tumors in plants through the TI, tumor-inducing. And the last one is restriction modification. Here, know that there are three. This is basically the safe sex in bacteria. You have three types of systems: type 1, type 2, and type 3. This is the one that we talked about most, because that's the one that cuts at palindromes. And the cell usually protects itself by methylating its DNA. So one question could be, let's say let's make it easy. Both, neither. So this is the last question. I'm giving you one strand of a double-stranded sequence. Okay, one strand of a double-stranded sequence. And ask you, potentially, type 2 will cut at this sequence, at that sequence, at both, or at neither. Assuming that it will cut. So does that make sense? So this is one strand of a double-stranded sequence. Does this make sense for it to be a side? Does this make sense for it to be a side? Both or neither? How can you tell? Type 2 cut at what? Type 2 cut at palindromic sequences. What are palindromic sequences in DNA? Sequences that read forwards and backwards the same way. So if this is GATC, what's the complement? GATC. Perfect. GATC, GATC. This is not so perfect. Because make the complement. TTCC has nothing to do with a palindrome. Only A will work. Well, that's what I had planned. If you guys have questions, please come up. And again, if someone is kind enough to donate their notes, or someone, I can post them for the class.
I could donate.
Well, if you want to, can you scan them? Right.
Yeah, I'll have
So
it
the
in the afternoon.
Right,
I'll just try it in the Marshall
Right,
Center.
right. Again, the more the merrier. If anyone else wants to. But thank you.
Awesome.
Well, it's in the makeup.
Oh, bro, you gotta text JP for you. You gotta text JP.
And the exam will be
Are you going to offer him the video, or is that for your personal?
Yeah, I can offer a video.
fill in the blank.
What's your email?
It's that.
There will be some of them that will be largely subjective.
Okay, so like, what are
It's probably, I can't tell you, but it's
Try
probably
type
happening.
and a short essay?
James.
No, right. I mean, if anything, it'll be small. So most of the essay would be, again, fill in the blanks.
Yeah, I'll try setting it up because they're both like 45 minutes or something like that. So,
So it's all the way
if
through?
I might just try to see if I could chop it up into smaller videos. Alright, awesome. And capital S?
No, it doesn't matter.
Okay, awesome. Alright,
Because it's going from RNA back to
no problem.
this transfer. This regular transfer. This regular.
No, that's like potato. What'd she say? Be scared.
Right. So capital transfer is removes the